Seed collecting can take a lot of time, effort, and funding as well. So it is important that we know how to treat seeds after they come back from the field and understand any limitations on storing seeds long term. In this session, we will focus on how to maintain a good quality collection through understanding seed air moisture relations, determining appropriate seed storage behavior, and some practical examples of processing seeds and assessing their quality. Hello everyone, my name is Aisyah Faruk and I'm the Conservation Partnership Coordinator for the Millennium Seed Bank Partnership. And today I will be guiding you through a very quick theory on seed drying. So by the end of the session, you'll understand why drying and freezing are important when it comes to long-term seed conservation. You'll understand how moisture content is affected by oil content, temperature, and relative humidity. And at the end, we will show you a video on how to determine seed moisture content using a hygrometer. Seeds have been an important development in the reproduction and success of gymnosperms and angiosperm. It is essentially the unit that is capable of developing into a new plant. The dispersal and germination of seeds have greatly influenced the spatial and temporal distribution of flowering plants. Obviously, there's a huge variation in the size and shape of seeds, with the cocodamia being one of the largest to the orchid seed, which can sometimes be microscopic in size. With all this variation, there is still a general structure for seeds. Here you can see a cross-section of one, and it's made up of three main sections. The embryo, which is essentially the thing that becomes the new plant. The endosperm, which is a form of nutrition, of stored nutrition that feeds the growing seedling. And finally, the seed coat, which is usually developed from the maternal tissue and can vary in structure from paper thin like in peanuts, fleshy like in pomegranates, or a hard shell. The function of a seed coat is to help protect the embryo from damage, which is something to keep in mind later when you start learning about dormancy breaking. So we know about the structure. Now we need to know a little bit about seed physiology, specifically how it interacts with water in the air. The first thing to know is that seeds are hygroscopic. This means that when the seed is drier than the air around it, the water in the air moves into the seed, like so. And this happens the other way around as well. When we have a wet seed, much wetter than the surrounding air, the water moves out of the seed and into the air. The greater the difference in moisture content between the seed and the air, the more readily the water will move. Eventually, a point is reached when there is no net movement of moisture between seeds and the air, and we call that the air and the seeds have reached equilibrium. At this point, the moisture content between the air and the seed is the same, so we say that the moisture content of the seed is at equilibrium moisture content, or EMC. And when the moisture content of the seed is at this equilibrium, we can also measure its equilibrium relative humidity, or ERH. A number of factors can affect the moisture content of seeds, such as the temperature and the composition of the seed itself. Oily seeds will have a lower moisture content at any relative humidity compared to its non-oily seeds. This is because, as we have seen, moisture content equates to seed's water content. If the space within a seed is taken up by oil, then the amount of water within that seed will be less, as oil and water do not mix. I can show this more clearly using this graph. This is the sorption isotherm for 4 different seed collection. which is essentially a graph showing the relationship between a seed's ERH here on the x-axis and its moisture content on the y-axis. As you can see, as the ERH increases, so does the moisture content. But it's not a linear relationship between the two. The exact shape of the isotherm depends on the composition of the seed and its temperature. For a given ERH, oily seeds, which are the green and black lines, have a lower moisture content than the non-oily seeds, shown here as red and blue lines. And this is because seeds with a high oil content will absorb relatively less water than their non-oily counterparts. When we look at the effects of temperature, we see that for a given ERH, here at 50%, the higher the temperature, the lower the moisture content. This is because water is more able to vaporize and diffuse out of the seeds at higher temperatures. Conversely, the lower the temperature, the higher the moisture content. So why are we so interested in drying? It is because of the impact drying has on seeds. For every 10% seeds are dry, their lifespan doubles. And for every 5 degrees seeds are cooled, their lifespan doubles as well. It is essential that you dry seeds thoroughly and monitor their ERH effectively, ideally using hygrometers. Reducing the seed moisture level slows down the aging process, prevents germination, it reduces the damage by insects and other pests, and more crucially, it enables seeds to be frozen. So what happens when a seed dries? When it dries, it goes through 3 different stages, which relates to how water is held in the tissue. These are known as water binding regions. The first one is called Region 3, which has an ERH of above 85%. Seeds at this ERH are generally fresh seeds where loosely bound water is still within the seed. This is water that is not related to any physiological activity. When a seed is at this state, normal cellular processes are happening, like growing. The water you find here, this free water, can freeze. So if seeds are frozen without removing this water, ice crystals will form and damage the cells. As you start to dry your seed, it reaches what we call Region 2, which has an ERH of around 15 to 85%. Some cellular processes take place here. The seed will actually keep on aging, but at a much slower rate. When you dry the seed further, you end up in Region 1, which is anything below 15% ERH. Here you start taking out strongly bound water. Seeds are in a relatively inert state, but do beware, further removal of water can actually damage the seed. We are looking for this ideal spot, which is around 15% ERH, where the seeds are still alive, dormant, and not aging. So in summary, seeds are hygroscopic, which means that they can exchange moisture with the air. When the two are at equilibrium, there is no more net exchange of moisture, and the seeds are at equilibrium moisture content. At this point, we can also measure the seed's equilibrium relative humidity. Moisture content of oily seeds will be lower than non-oily seeds at the same relative humidity, and is also lower at higher temperatures. Drying and freezing can increase the longevity of seeds, and it is important to dry seeds before freezing, although do beware of over-drying. In the next video, my colleague John Adams will run through how to use a hygrometer to measure the equilibrium relative humidity of seeds.
Drying is one of the most important factors in maximising seed longevity. But how do we know if a seed is dry? Traditionally, the technique was called gravimetric moisture content, where you took a sample of seeds, you weighed it, placed the sample in an oven at an elevated temperature, left it overnight, and in the morning re-weighed it, and the difference between the two weights was the moisture lost. This is obviously a destructive technique and takes a long time to do. An alternative method is to use equilibrium relative humidity. Relative humidity is the amount of moisture held in air. Equilibrium relative humidity is the amount of moisture in the air above a sample. So in this case, you see you have a container of seeds, and these seeds will either lose or gain moisture until they are in equilibrium with that air. If you then measure the relative humidity of that air, you then have the equilibrium relative humidity of your seed sample. The advantage of equilibrium relative humidity testing is that it's very fast, typically 15 to 20 minutes, and it's non-destructive, which is particularly important for small, valuable seed collections and threatened species. To ensure an accurate reading using the ERH technique, you need at least 20% of your volume full of your seed sample. So the way we measure the relative humidity is to use a hygrometer. This particular model has a small probe here where there's a sensor in the end for both temperature and humidity. If you place your seed sample in a small container like this one, then gently insert the probe into this container, having sealed the sensor in with your seed sample, making sure there's a tight fit, avoiding holding the sample container because it will warm up in your hand. You can now leave this for 15 minutes or so, or until the reading stops changing and you get an equilibrium relative humidity reading. Because the term relative humidity is, as it says, relative to a particular temperature, it's always important that when you record an equilibrium relative humidity level, you also record the temperature at which that has occurred. This type of hygrometer not only can be used for measuring seeds, as we see with a sample container, but also can be taken into the field to actually measure the air and the ambient conditions. This hygrometer also has the ability to data log readings. You can program this to take readings every minute, every hour, or every day. Set this up and then place this in your collecting bag of seeds. Close this up, put that in your dry environment, and this will actually monitor the drying rate of that collection of seeds. This is an alternative set of hygrometers attached to a laptop. The laptop records the readings and represents it as a graph. By equilibration, we mean when the actual readings stop changing. So here you get a level line. So now you know how to measure the ERH, or the moisture status of your seeds. And this is important. For every 10% you can reduce the relative humidity, you can double the seed's life.
Hello again, and welcome to the next lecture in the session. Here we will go through seed storage behavior. As you're already well aware, seeds of wild species come in a range of different forms. They are also varied in how they can behave in storage. In this section of the lecture, we will explore the three main categories of seed storage behavior. You'll understand their relevance to long-term seed conservation, and some practical examples of how to identify and manage for different storage behaviors. The storage behavior of seeds come in three main categories: orthodox, intermediate, and recalcitrant. However, in reality, the desiccation tolerance of seeds actually sits along a gradient, which is shown here. Orthodox seeds are those that can withstand drying and subsequently can be frozen to increase longevity. Around 80% of known seed-bearing plants have orthodox seeds. Intermediate seeds are those that can be dried to a certain extent. Some of those can actually be dried to the appropriate ERH for freezing. But these sometimes showed a loss in viability, sometimes dramatically, when banked using conventional methods. Recalcitrant seeds are those that cannot be dried below 90% ERH and therefore cannot be frozen in conventional seed banks. There are some options for storing some intermediate and recalcitrant seeds, one of which is moist storage, but their longevity at this stage is limited and only done for short-term storage. This option will also require very close monitoring, as if the seeds are too wet or too warm, they tend to germinate. If they're too cool, they can suffer chilling, and there is also the risk of fungal contamination. The second is cryopreservation. However, this method can be quite challenging as it requires species-specific protocols in order to be successful. So how do we know if we've got recalcitrant seed? We can use a number of different methods, which we can go through right now, including looking at the taxonomy, scouring the literature, predicting our seed traits, and also some laboratory experiments. So let's start with taxonomy. Desiccation sensitivity has been recorded in around 65 different plant families. Some families have a higher frequency of species with recorded recalcitrant seeds than others. For example, very high levels in Dipterocarps, with 98% of the species showing desiccation sensitivity, but extremely low in Poaceae, for example, down here. There are also a few families where we can't really tell for certain. For example, the Ericaceae, where we have around the same percentage of orthodox and recalcitrant seeds, with a third of them we're not too sure about in the first place. So although this method can be used in quite a number of situations, there are others where I think a little bit more work is needed. We can look at the published literature. This is the compendium of information on seed storage behavior. It lists the seed storage behavior of around 7,000 species. And Q Seed Information Database has actually been built upon this compendium dataset and has now included storage behavior records of around 18,000 species, probably more since this recording has been made. You can see the link to the actual tool on below. Let me just get my pointer. This is the link to the tool. And you can choose, you can filter out your family, genus, species using the information database, and it will tell you whether you've got recalcitrant or intermediate species. These are some figures we extracted from SID. You can tell that because we've been in the conventional seed banking business for a while, we have quite a bias towards drier regions. So we've got quite a lot of orthodox species here. But what do you do if your target species isn't in SID and the family that is in doesn't really show, you know, one way or another? If we know the species' physical attributes and a bit about its ecology, we can actually use a prediction to see how it will behave in storage. A seed's physical attribute can be a way of quickly determining whether a seed is recalcitrant or not. Seeds that are recalcitrant or desiccant sensitive tend to be large with thin seed coats and have a 1,000 seed weight of more than 500 grams. They also show to have an initial moisture content of about above 30% ERH. There are some who say the absence of an endiosperm or the presence of a green embryo could also indicate recalcitrant seeds, but this hasn't really been studied conclusively yet, so we can't really verify it. This is a paper by Tweddle and company in 2003, where they looked at where the woody species are found and their seed storage behaviors. You can see through this graph that as the climate becomes drier, the percentage of species with recalcitrant seeds goes down. So woody rainforest species, for example, are more likely to produce desiccation-sensitive seeds, some even up to 50% more likely, while woody savanna species are more likely to produce desiccant-tolerant species. In some instances, knowing the seed dispersal behavior of your target species can give you a bit of a clue over its seed storage behavior as well. Here we have 4 tree species found in the African drylands. 2 of these species shed their seeds during the wet season. If you can see here, the bar graph shows rainfall, and the arrows are where these species actually are known to shed their seeds. And these species actually have desiccation-sensitive seeds. In comparison, these on the left-hand side shed their seeds where rainfall is very minimal, so the drier months. And these 2 species have orthodox or desiccation-tolerant seeds. Building on the use of taxonomy and traits to predict seed behavior, Wise and Dickey published a paper in 2017 outlining a model that can be used to predict the likelihood of a particular species having orthodox or recalcitrant seeds. They incorporated the known behavior of the target species' closest relatives, where it comes from, so its habitat and climate variables, and any of its specific traits, for example, woodiness or dispersal modes. Based on their model, they predicted that 8% of species globally are recalcitrant. But when we go into the tropics, their estimate actually jumps to 18.5%. This is a protocol developed by Hong and Ellis to determine seed storage behavior. It is a very comprehensive test. Basically, this involves an initial determination of moisture content and viability when the seed is fresh. This is then followed by desiccating the seeds to an intermediate moisture level at around 10 to 12% ERH. Sorry, 10 to 12% moisture content. Seeds that don't germinate are probably recalcitrant. Another subsample of seeds are then further dried to 5% moisture content, and this is then tested for viability again. If they don't germinate, they're probably intermediates. The final subsample of seeds are dried to 5% and stored at -20 degrees C. If they don't germinate after this, they're most likely intermediates, but if they do germinate, then they're orthodox. I said that this is very comprehensive, and it is. The problem with this method is that you need thousands of seeds to be able to do it properly. It is therefore rather unlikely and unrealistic for many of our partners. This is an alternative that was developed by Pritchard in 2004, and it's called the 100 seed test. At first, you'll still need to do the initial moisture content on a subset of seeds, then you germinate the fresh seeds. At the second stage, you will have to dry 32 seeds by mixing them with an equal weight of silica gel in a sealed container. You will need to change the silica gel and weigh the seeds every 1 to 3 days until the seeds have reached a constant weight. This means that the seeds are at equilibrium. At the same time, you will have a control group, which is the same number of seeds in a high humidity sealed container. You can use anything like moist vermiculite or filter paper, for example. It's just to maintain the humid conditions. You will then split each set into subsets, one subset used to determine moisture content and the other for germination testing. This method uses much less seeds, but it also only gives you a limited number of results. For example, you won't be able to distinguish between recalcitrant and intermediates using this method. This is what the 100 seed test results might look like. You'll have a graph with percentage germination on the y-axis and time on the x-axis. If your seeds are desiccatant tolerant, you will have the curve that resembles one on this side. You'll see that the shape of the fresh seeds, the moist store seeds, and the dried seeds have the same pattern. If you have seeds that are desiccation sensitive, the curve for the fresh seeds and the moist stored seeds will remain the same, but the subset of seeds that are dried will not germinate at all. Note that I've said desiccation tolerant and not orthodox or recalcitrant. Again, another limitation of this test is that it does not have a step that stores the seeds at -20 degrees C after drying, and you can only call the seed orthodox if it is able to tolerate desiccation and storage at sub-zero temperatures. With all the caveats of the 100 seed test, it's still a very effective tool for guiding seed conservation, particularly for woody species or those with small collections. In 2019, Montana and colleagues set out to assess seed desiccation responses for native trees of the Caribbean. You can see here the number of seed lots that they have to work with is very limited, and therefore would not be suitable for the comprehensive method. However, they found that 80% of the seed lots from the 40 species that they were studying showed consistent results among experiments using the 100 seed test and predictive models. The study also highlighted specific knowledge gaps for species that required further investigation. The 100 seed test has been widely adopted within the MSBP and cited by partners in the UK, Brazil, Burkina Faso, Australia, Madagascar, and the Dominican Republic, so it is widely used within our partnership. So in summary, seed storage behavior is a continuum from orthodox to recalcitrant. Only orthodox seeds can be conserved in traditional seed banks. Intermediate seeds can only tolerate partial drying and also lose longevity within -20. Recalcitrant seeds cannot be dried and therefore cannot be banked, We can predict a seed is recalcitrant through various different methods, including standardized experiments to confirm their storage behaviors. This is particularly important for regions where there are a lot of unknowns. Thank you for listening. The next lecture will be on seeds processing and quality assessment of collections, and this will be taken by Lucy Taylor.
Hello and welcome to the Seed Processing Techniques and Quality Assessment lecture. I'm Lucy and I work as a Seed Collections Assistant at the Millennium Seed Bank. So by the end of this presentation, we will become familiar with the different stages of processing seed collections. We will gain an understanding of basic seed cleaning techniques and how seed quality is assessed. And we will also become aware of some potential issues you may face during seed processing. So when we think about cleaning wild seed collections, we need to be aware that there's huge variation in seed and fruit size. There's also variation in robustness, etc. So we're going to need a number of different processing techniques, and each individual collection should be treated as an individual. Quite often, if the seeds or fruits can be dried before processing, the cleaning is easier and less likely to damage the seed. Or we need to clean to a reasonable end point. It's important to realise that you might not always get a perfect collection with a wild seed collection, and it's extremely important that we do not lose or damage any seed. So in terms of health and safety, it's important to follow good lab practice and wear a lab coat. Treat all seed collections as potentially poisonous or irritant. If you don't know what the species is, assume it is poisonous or irritant. Many of the techniques we use produce dust, so either wear a mask or work in a dust extraction hood. Make sure you dispose of all unwanted plant material, dead insects and disposable gloves carefully. Clean all your working areas, including the floor and equipment, carefully after each collection is processed. This is to avoid cross-contamination. And when you've finished, remove your lab coats when leaving the cleaning room and wash your hands thoroughly. So before you begin cleaning, it's important to carry out a visual examination. So examine the fruit and seed structures to help determine the best techniques for you to use. So before you begin, you need to work out, is it a seed or a fruit? Is there any contamination with any other seeds? Are there any obviously empty or infested seeds? Do you have a range of maturities amongst the seeds, or are they all the same? Is there any morphological variation? And will the seeds be easily damaged or lost during cleaning? The best thing to do once you've chosen your technique is to try it out on a small portion first, before expanding it to the whole collection. So before you begin cleaning, it's important to make sure you know exactly what the seed is. Here is an example of Taranthus Dichotomous from the Caryophyllaceae family. Now you might think the seed is the large brown triangle shape at the bottom, but actually the seeds are much smaller and are hidden inside the spines. If you weren't careful and didn't know exactly what you were looking at, it would be very easy to lose the seeds from this collection. So we're now going to show you some of the techniques available for processing and cleaning seeds. We're going to show you these in much better detail in the following video on seed cleaning techniques. So for now, I will just show you some pictures. The first technique we have available is sieving. You can also adapt the sieving technique to use a rubber bung for seeds that are a bit more robust. The sieve and bung technique can also be adapted to process fleshy fruits. It's important that fleshy material should be dealt with immediately to avoid losing seed viability. It's also important to use warm, not hot water, as hot water may damage the viability of the seeds. If you have to get the seeds wet, the best way to dry them is to spread them out in a thin layer on a nylon mesh with a drip tray below to catch the water. Gloved rubbing is a more gentle technique than sieving and bunging. It's also very effective with fluffy or fragile seeds. Aspirators are useful machines that separate items based on weight, and they use an airflow to do this. So empty seeds or chaff will weigh a lot less than full seeds. If you don't have access to an aspirator, you can try some more traditional techniques. If you think you have a technique that might work, it's good to give it a go, but make sure you aren't damaging any of your seeds. Bag crushing can be used, but make sure you have a good pair of gloves. Foot crushing can also be used, but check to make sure you're not damaging any of the seeds. Capsule shaking is a very easy technique and helps prevent debris. Hand sorting is a very accurate method, but it can take a very long time, so decide if it's worth it before you start. Some seeds are covered in a sticky or oily substance, which can make it much harder to process. If you coat the seeds in a fine powder or ash, this will help absorb and remove the excess oil or mucus. If you're going to use talcum powder to separate the seeds, make sure it's unscented due to the chemicals in it. If you're going to use ash, it's not harmful to the seeds and it will not damage your equipment, and in the case of oily seeds, will absorb the excess oil. The oil-soaked ash can then be rubbed off in a sieve or with a cloth, leaving clean seeds. So here we're going to look at some of the issues you may come across when cleaning seed collections. You may find that within your collection there's a range of maturities. If some fruits or seeds appear to be immature, these could be separated out and allowed to mature further under ambient conditions. You may find, particularly in families like the Apiaceae and Asteraceae, that there's morphological variation within one species. You need to make sure that you know exactly what all the seeds look like and that your chosen seed cleaning technique doesn't remove a particular variation, because you want to keep all the genetic variability. If you come across cones or similar that need fire to open them, make sure you hold them away from you using metal tongs and hold the cone over a container to catch any loose seeds. Carefully treat the cone with a blowtorch, avoid heating the insides of the cone scales and do not overheat any one area. When the cones are open, allow to dry so they continue opening, enabling the seeds to fall out. Once you've chosen the appropriate technique to clean your collection, make sure you always perform a visual examination before you begin. Cut test a few seeds before, during and after cleaning to assess the quality. Process a small sample to completion before working on the entire collection. Save all of the field seeds and clean all equipment and work areas between collections to avoid cross-contamination. Once you are happy and have finished cleaning your collection, we then need to assess the quality of the seeds. One of the techniques you can use is X-raying. You can see the picture on the left. All the seeds look fairly similar from the outside, but once you do an X-ray, you can clearly see some of the seeds which are bad, and we would probably call them empty. So X-raying images to assess the quality of a seed collection does have some advantages. It's relatively quick and it's also non-destructive. The images can also be saved for future interpretation. The disadvantages are that it cannot be used for all seeds. Seeds in the Scrophulariaceae and Cyperaceae families cannot be interpreted clearly on an X-ray image. The X-ray seeds should also be kept separate, just in case X-raying them has impacted them in any way. Seeds within fruits may not show up clearly on the X-ray image. X-raying is also not a measure of viability. It can give you an idea of the quality, but it cannot tell you if the seeds are alive. An alternative to X-ray analysis is cut testing. Now, cut testing is a very good way of assessing the quality of seeds, but it does take some time as you have to cut each individual seed and examine each contents. Cut testing allows you to identify empty, poorly developed and infested seeds. It can also give you important clues to germination requirements, i.e. it can give you an idea of how big the embryo is, where the radical is and where it will emerge from the seed. The downside of cut testing is that it's destructive. Once you've cut the seed, you've killed it, so it reduces the size of the collection. For some seeds, it's also quite difficult to cut them safely. So one of the things we have designed is on the picture on the right. It's a clamp with a section cut through it, so we can easily and safely hold the seeds while cut testing them. So to cut test seeds to get the quality, we need to randomly sample 10 seeds and cut them lengthwise from end to end so you can see as much of the content as possible. We need to record the number of seeds which are full. These are firm and often have white tissues. Part full. Now these don't look quite as good as a full seed, but you think they'll still be able to germinate. Empty seeds, which contain little or no seed tissue, and infested seeds. Look for evidence of predation such as an insect exit hole or for insect frass. So why and when do we need to cut tests and X-rays? Well, previously you will have now covered points 1, 2 and 3, and we will cover point 4 in future lectures. For point 1, we need to cut test at the point of seed collection. We need to assess the seed maturity and quality before we begin to collect. We need to cut test during cleaning to check that the cleaning process is not damaging the seeds and that good seeds are present. We need to cut test or X-ray after cleaning to assess the initial seed quality. We record the initial seed quality on the accessioning card, and we use this to calculate the adjusted seed quantity and the oversow in the germination test. We also cut test after the germination test. This is to assess if the ungerminated seeds are fresh, mouldy, empty or infested. Most collections of wild seeds will include some empty and infested seeds. It's not always possible to remove these at cleaning. The adjusted seed quantity uses the results from the cut test or X-ray to estimate the number of full seeds in a collection. To do this, we need to work out the proportion of good seeds. We do this by taking the number of full plus the number of part full seeds in the cut test and divide it by the total number of seeds used in the cut test. We can then work out the adjusted quantity by taking the current quantity and timesing it by the proportion of good seeds. For example, if we have 10,000 seeds in a collection and the cut test or X-ray show that the proportion of good seeds was 0.9, then the adjusted seed quantity is 10,000 times 0.9 equals 9,000 seeds. We need to count the number of seeds so we can best plan the future use of the collection. To do this, we weigh 5 samples of 50 seeds and then we weigh the remainder of the collection. You can now calculate the average weight of 50 seeds, then the average weight of 1 seed. You can calculate the current or the total quantity of seeds, and you can also calculate the adjusted amount of seeds, i.e. the number of good seeds. Now, if the seeds are very small, weigh 1 sample of 250 seeds. It's important to use a random sampling technique, and if some debris remains after cleaning, include it in the samples. This picture shows some of the basic equipment you need to count seeds. You can make a simple counting card, shown in the bottom left, by folding a laminated piece of cardboard. This makes counting a lot easier. So when we think about seed banking and long-term storage, we need to ensure the dry seeds are efficiently packaged for long-term cold storage. We need to ensure the container is secure, the seeds remain dry, any X-ray samples are kept separate, and the label contains the serial number and the location. The picture on the right shows a tri-laminate foil bag, which we are now banking most of our seed collections into. We previously used glass jars, but we now believe these foil bags are a better long-term cold storage solution. So there are some practical considerations when it comes to banking. How are you going to test the moisture content before banking to make sure the seeds are dry enough? Here at the Millennium Seed Bank, we've designed these special rotronics, which you can see on the left. You need to think about awkward seeds, for example, spiky seeds, seeds that are very small, seeds that are very big. How are you going to store them safely? With small seeds, you may also find that static is a huge problem and that they stick to everything. The bottom right-hand picture shows an anti-static device, which can be very useful. So to summarise the seed processing key points, we have now become familiar with the different stages of processing seed collections. We have gained an understanding of basic seed cleaning techniques and how seed quality is assessed. We have also become aware of some potential issues you may face during seed processing. We will now play a video showing the seed cleaning techniques.